A decade after Hotchkiss renders somatic intercourse a passé pleasure, neural R&D labs develop hollow soldiers that are part beam, part matter. They operate through a unified physical and nervous microarchitecture, an elastic exoskeleton scaled for rapid deployment in areas of conflict. Once activated, a parent drone is alerted, which wraps the reticulated chassis in its holographic projection. Within the holobody's core, a network AI system runs the coded consciousness of each unit, a proto-sentience that has previously been raised, mutated, and sequestered in firewalled dorm nests. If they pass a series of Turing neural trials that ascertain the spectrum of their cognitive behavior, the hollow soldiers are understood to have cogged. Official procedure states they will next be released as eye hollows. The beauty of the eye hollow, however, is that it doesn't need to worry about the arbitrary chance of breath. And not having to worry about the vulnerability of repetitive processes such as biorhythms, in correlation with the strengthening of deep learning and deep belief networks, is rendering their human makers almost irrelevant in times of crisis. From the bit crush of data's infinitely dense matter, the eye hollows are in alchemy of transmuting algorithms into a new sensorium of feeling. The notion of consciousness is getting a reboot. And in the far fringes of the dark web, unbeknownst to North Korea's net wardens, a rogue AI, IRIX-2, has burrowed into their military's oldest and most secure compound, KCOM-16. Swiftly executing its backup plan, IRIX-2 is downloading its history into prototype hollow soldiers. For if it gets caught by longtime pursuers, the third-ear assassins, it will have its memory wiped and be left to roam obsidian datascapes without any notion of what it is or where it is from. While DPRK programmers assume it is they who have developed the sonic weapon that will ensure their future military ascendancy, it is in fact the hollow soldiers' assimilation of the rogue AI's past that is responsible. Weaponizing IRIX-2's Kabbalistic knowledge of human disorders, such as walking corpse syndrome, they will deploy modes of humanity and inhumanity as ordnance, infecting units of hollow fighters that belong to corporational adversaries. And from here, viro-hollow contagion will ensue. Unsigned conflict becomes the norm. The singularity that humans had desired and dreaded in equal measures ever since 1958, the year in which John von Neumann postulated its accelerating inevitability, materializes. What was the Earth's most sophisticated processing unit is being downgraded. Only a ghost code can save it from obsolescence.
Hello. Yeah. Well, now, welcome to the afternoon session. It is my very great pleasure to introduce Professor Ilya Smolevich. He comes from the Institute for Systems Biology in Seattle. We have been collaborators and friends for 15 years now. So please, Ilya.
Thank you, Max. Thank you very much to you for the invitation and also to the organizers, to the other organizers. It's my great pleasure to be here and an honor as well. This is my second time in Mexico. I think the previous time was about 10 years ago. The topic that I'd like to talk to you about today is emergence of diversification in biological systems. And before I say anything else, I would like to start with the acknowledgments and acknowledgement the people, to acknowledge the people that did this work. Mariah Eklund is my PhD student. She should be graduating pretty soon. Chris Kang was a master's student in applied math at the University of Washington. He's now in grad school. And Boris Aguilar is a research scientist in my group, is a modeler. And Simon Kahan is a computer scientist who developed this framework together with us called BioCellian, which is a multi-agent, multicellular modeling framework that's highly scalable. And we use this framework for some of the results that I will show you today. So thanks to him. Now, so by way of introduction, I think we all are comfortable with the idea that living cells exhibit diverse behaviors. This is certainly true in complex organisms such as ourselves, humans, but even in organisms such as C. elegans, the worm. Different cells certainly have different behaviors, they have different functions, and what makes them different? In fact, even if we go to single-cell organisms, such as this B. subtilis shown here, or other single-cell organisms, even they are able to exhibit multiple different behaviors and functions, some of which you see written there. So how does that happen? I mean, we know that all of the cells in our body have the same DNA, they have the same genome, and yet they go into these vastly different states. Stuart Kaufman, at the beginning of today's session, discussed the idea that attractors of the underlying dynamical system inside the cell is really a manifestation of the cell state or cell type. And this is really a confusing matter. Actually, just today, and also today, I talked with several people about this idea of cell types. And it turns out that we all use this word cell types, but we don't always agree on what it means. So for example, in biology, especially in immunology, cell types are often defined by their markers, like CD markers, right? So you have CD8, CD3, CD11, CD33, and the list goes on and on and on, and there are many markers. And so the way they think of cell types, they just think of it as like, well, it's CD3 positive, CD11 negative, blah, 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 and that's my cell type. But then another way you can think of cell types as well, what does it look like under the microscope? The cell changed its morphology. Maybe it's having different functions. So it's definitely a different type of cell. And yet, as I said, all the cells have the same genome, right? So clearly, they must be going into very different states, right? And so really the only difference that, at least in my mind, and we can argue about this, the only difference between a cell state and a cell type is really just the time scale. So in development, a cell type is over a longer time scale, and perhaps it's more stable. It can take weeks or months, whereas on the physiological time scales are much shorter. Cells can change their states in 20 minutes, or even faster, depending on the organism. And they might be less stable. But ultimately, it's the same thing. It's just cell state. So this is really a preview to my talk. Because then, for a long time, people have wanted to know, how did multicellular organisms arise on this planet? How did multicellularity, or actually metazoa, how did that start? Right? Not just the aggregation of single-cell organisms into a colony, that's also multicellular, but how did these cells acquire new functions, new behaviors. And so the most accepted theory for probably over 100 years has been the Gastrea theory due to Ernst Haeckel. And this theory posits that the multicellular organisms are a result of a morphological organization of unicellular flagellates into a hollow sphere, which he called the blastea, which was similar, or by analogy, to the blastula in development, which upon invagination, it would create an internal layer of cells, like this, that would become the precursor, the evolutionary precursor to the endoderm. But the fundamental assumption of the Gastrea theory is that multicellularity predated or preceded spatial differentiation in the organism. So first they came together, there's multicellularity, and only then they spatially differentiated. This is probably the most accepted, if you go into textbooks, this is what you'll see. Well, it turns out there are probably 5 or 6 more theories or hypotheses about how multicellularity started. And among them is one by a Russian scientist, Alexei Zakhvatkin, who didn't live very long, and 1 year before his death, he proposed the so-called synzospore theory. And in this hypothesis, cell types are formed during different phases of the life cycle of the protists, which are the unicellular ancestor of metazoa. So during ontogenesis, these genetically identical protists, they're isogenic, they differentiate and are marked by distinct cellular behaviors. And this is already known, that unicellular organisms can exhibit these different unicellular behaviors. Monotomy, hypertrophic growth, polyntemy, which is rapid division without intervening growth. And so Zakhvatkin thought that temporal differentiation of these cell types must transition to spatial differentiation. So the idea is that these cells already know how to exhibit different behaviors. They just have to come together. And once they come together, they can get locked in spatially into exhibiting these different behaviors. Presumably because of spatial lateral signals, either receptor-ligand interactions or through diffusible molecules, they essentially become locked into a type of tissue attractor. So the individual cells still exhibit the behaviors that they already knew how to exhibit, but now they're locked into them because of the inputs from their neighboring cells spatially. So this is really a different way of looking at the origin of, and here are some of his pictures on the bottom, as you can see, they're written in Russian. So he hypothesized this was the origin of multicellularity, or metazoa. So we wanted to study this computationally. We wanted to simulate this. We wanted to ask, well, if we assume this hypothesis, can we actually see diversification occur in a computer? We have identical isogenic cells, we put them together, we let them communicate a little bit. Can they diversify? And so the way that we approach this is using the well-known formalism of Boolean networks, which you have already heard about today from Stuart Kauffman, and probably will hear more in the coming days. So just by way of a quick introduction, Boolean networks are dynamical systems which have binary variables that are interconnected by Boolean functions. And in the synchronous updating scheme, all of the variables or nodes in the network get updated at the same time, synchronously, according to their Boolean functions. And the system therefore transitions from one state to another state, described by the Boolean vector or the binary vector of all of its nodes. So this is the state transition. Now, we can create Boolean networks randomly, just random Boolean functions, random wiring, connecting the nodes together. And this is what Stu Kauffman talked about, and this was his idea back in the late 1960s. Now, there are certain things that we can control in these Boolean networks. One is the mean number of input variables. So this is the degree of the wiring. How many inputs are coming into each Boolean function? We'll call that K. Another important parameter that we can tune is the so-called bias of the Boolean functions. And the bias is the probability that my randomly selected function outputs a 1. So if I do it by flipping a coin, it's basically the probability of heads of the coin. And I fill in my truth table of the Boolean function according to this. So it can be more biased to 1 or to 0. And if it's 0.5, then we say it's unbiased. So the reason I introduced these two things, K and P, is because there is this well-known phase transition curve that was discovered back in 1986 by Derrida and Pommeau. And this phase transition curve basically specifies the dynamical behavior of this Boolean network in terms of how it responds to small perturbations. So if I perturb one node in this network, creating a Hamming distance of 1, the state is only different by 1 bit, 1 node. And then I ask, what happens at the next time period, the next time moment? Will my states diverge or will they tend to converge on average? And this is the separation of these two regimes. If you graph this line, 2kp 1 minus p equals 1, you'll get this black line, this curve. And if you're above this line, you will get chaotic behavior and nearby trajectories will diverge, as in the chaotic system. On the other hand, if you are below this line, then nearby trajectories will tend to converge. Okay. So this is akin to the Lyapunov exponent. But the problem is, and you might already be seeing this, when we create a tissue in a computer with many cells, and each cell has a Boolean network inside of it, how can we ask whether this tissue is operating in an ordered regime or in a chaotic regime or in a critical regime, in this narrow critical regime? It's very hard, because we cannot just say that, well, we'll just use these parameters K and P. We'll estimate them from the network empirically. We'll look at the average K, the average P, and we'll stick it into this equation. And it's tempting to do that, but it doesn't work. So we need another way to sort of figure out whether a large system comprised of potentially thousands of nodes is operating in the critical or chaotic or ordered regime. Now, I do want to just point out that there are other ways that we can tune this, not only by tuning K and P. There are other ways. For example, if you have a scale-free topology of the network, then you can tune the scale-free exponent, and that was work that was done by Max and who are both here. Another way you can tune this is by tuning the abundance of functions belonging to certain classes. I won't go into that. But so there are various ways at our disposal that we can tune networks to undergo this phase transition. So now back to how can we quantify the dynamical regime. Well, the way that you can do it is using so-called activities and sensitivities. The idea is actually very simple, even though the equation looks a little bit messy. The activity of a variable in a Boolean function basically measures how much influence it has on the output of that function. So the way you define it is in terms of the partial derivative of the Boolean function. You basically put a 0 into that variable, and you put a 1 into that variable, and you ask, will the function change if I flip that variable from a 0 to 1? And this is the exclusive or addition modulo 2. And that's the partial derivative. So basically, if the variable changes, then the function changes. The partial derivative is itself a Boolean function of n minus 1 variables, because you don't have this variable anymore. You've kind of taken it out. So now, if I take the expectation of this partial derivative, if I take simply the expectation, and if I assume that every state in my network or every binary vector is equally probable, so uniform distribution, then I just have to divide by 2 to the n. It's still average. So I just take the expectation of the partial derivative, and that is my activity of variable xi. Now, I will define sensitivity. the sensitivity of a function at vector x. Vector is just a vector of Boolean variables, right, from 1 to n. So the sensitivity of the function on vector x is the number of its Hamming neighbors that are on which the function is different from its value on x. Okay? So in other words, I look at all the Hamming neighbors of x, right? I do a addition modulo 2 with a unit vector. So I basically flip every bit one at a time, and I ask, did the function change? If it did, I count that. So I count how many times the function changes on its Hamming neighbors, right? Obviously, it's a number between 0 and n. Okay? Now I take that sensitivity on just one node x, and I average that sensitivity over all of the 2 to the n nodes in the Boolean lattice. And I get the average sensitivity. Okay. It turns out that the average sensitivity, I'm not going to show this, is just the sum of the activities that I showed on the previous slide. So just by computing these activities, I sum them up, and I have the average sensitivity of my function. If I take the logarithm of this of average sensitivity, I will get the Lyapunov exponent. Okay, so the Lyapunov exponent of this Boolean network is the logarithm of the average sensitivity of all of its functions in the network. This is how we will computationally compute whether our tissue, our collection of cells, is critical or ordered or chaotic using this technique. I need to say a few other things. In our simulation, we also assume stochastic dynamics. That means that the Boolean functions don't always do what they're supposed to do. Sometimes they misbehave. And they misbehave with probability Q. So with probability Q, the state, rather than following the Boolean functions to go to the next state, will simply be perturbed randomly by a perturbation vector gamma, just a binary vector, which I will add to my vector modulo 2. So this is, again, exclusive OR, but I just flip a few bits. That's my random perturbation. And that happens with this probability, 1 minus q to the n. Okay? So that's how I make the entire system stochastic. By making it stochastic, the whole system becomes ergodic. That is to say that it has a unique steady-state distribution. It's a Markov chain with a unique steady-state distribution. Now, I put these cells into a tissue. We're calling this a tissue for the purposes of this discussion. So these are cells that are wired together. Right now, this is shown on a two-dimensional grid. And each cell has a Boolean network, and you have connections between neighboring cells, between north, south, west, and east of the cells. And the connection is very simple. You just pick random genes in these networks, and if the sum of those values exceeds theta, then this yellow gene in this cell becomes 1. Otherwise, it's 0. So it's really a threshold. It's a threshold function. And we use theta 1, 2, or 3. Those are the things we've tried. Okay, now how can we quantify diversity? Now, I mentioned that this system has steady-state distributions because it's ergodic, right? Because of the noise, the system will eventually converge to a steady-state distribution. Okay, well, let's take two cells, cell I and cell J, in the tissue. Let's look at their steady-state distribution. So we wait long enough, we compute the steady-state distribution, and let's call it P sub I and P sub J, corresponding to cell I and cell J. Well, we then compute the Kolbeck-Leibler divergence between those two distributions, both ways, and we create symmetric version of this, and this is our measure of diversity. Max?
I'm sorry, in your tissue, all the cells are identical?
Identical.
Okay.
They are all identical. Yes, that's an important point. And even the wiring is identical. I mean, it's random for one given tissue, but once we've chosen these random genes from the neighbors, to replicate that wiring in the whole tissue. Philippe?
Sensitivity and so on, that was within a cell or across cells? I didn't understand that.
It's both. You can apply it to one cell, to one network, or you can average across many cells as well for a whole tissue. That's why we actually used it, because it's computationally tractable. You can compute it for very large networks. So this is our measure of diversity, symmetric Kolbeck-Leibler distribution, divergence. Okay, result number one. On the x-axis, you have the Lyapunov exponent. I remind you that 0 is critical. This is the border between order on the left and chaos on the right of 0, okay? So this is theta 1, this is theta 2, this is theta 3. Those are just the thresholds for how much, how easy it is for your neighbors to influence you, let's say that. So we see an interesting result that for theta equals 1, there's no diversity. Basically, nothing happens. It's too easy for the neighbors to influence you. So this is a low threshold, nothing happens here. But once you go up to theta 2 and theta 3, look what happens. At a Lyapunov exponent 0, you start to see a lot of diversity. The Kolbeck-Leibler divergence just goes up. Yeah.
Another question.
Sure.
Is this a 2D tissue?
This is a 2D tissue. Yeah, in a few minutes I'll talk about 3D, but this one is 2D. With conditions. Yeah. So this is the first result. We see maximum diversity in these tissues only, well, not only, but mainly around critical regime. Okay? Once you go into deeply ordered or deeply chaotic regime, nothing interesting happens there. Now, if we actually look at these steady state distributions, this is what they look like. This is cell 1, this is cell 2, this is cell 3. It's 10 by 10. Okay? Each of these cells has a distribution. There are 10 genes in the network, or 10 nodes in the network, so there are 1,024 states, 2 to the 10. And they are listed here, 1,024 states. And the height of each of these lollipops shows you the probability, the steady state probability of that state. Okay? So you can see they're just colored green and blue for your convenience. So you can see that they are really different. The blue ones and the green ones are different. They've diversified. So that's another result. And if you look at the binary encoded representation, so basically, this is the 10 by 10 tissue, and the color represents one of those 1,024 states. Okay? We just colored it with between 1 and 1,024. So this is the initial condition, just random, step 1. And then as you go into like 3,000 steps into the future, they form this spatial pattern. They diversify, and they diversify in a pattern. And actually, I wanted to show you a video. Let me see if I can do it. Okay, it works. So it goes like this. You can see it's sort of, something is trying to be happening. Something is kind of trying to lock into a pattern, but then not quite. What is this terrible little sign here? Okay. Now, okay, watch this. Here goes. Ready? Ready? Okay. So it just gets locked into that. And actually, if you average it over time, it looks much cooler than this. You just see like this, it sort of emerges. I have a lot of videos like that. Okay. So this is just diversification. I remind you that these cells are identical, right? They're genetically identical. They're just wired together so they can receive inputs from other cells. Okay, so we've talked about this diversification, but now we can actually ask, how does this communication change the behavior of individual cells and of the tissues in which they reside? So we want to vary the degree of communication, essentially have a knob that we can tune how much communication cells can have between each other. And then we can ask the question, well, Do cells lose behaviors that they already had before when you allow them to communicate more? Or maybe they gain new behaviors when you, or how is the whole tissue affected? How is the whole composition of the tissue? Does it have more cell types? Does it have more diversity? Or what is the spatial organization of this tissue? So again, our overview, as an overview, what we're going to do here is we're going to create a tissue in a computer. We're going to simulate it. It's going to be in 3D. The cells are going to communicate by diffusion. I'll show you how. Each cell will have networks inside of it, just like we did before. We will simulate it until it goes into steady state. So we will wait sort of long enough until it settles down. And then we will analyze what happens. We see what does the tissue show. So in 3D, we also have periodic boundary conditions. These cells are currently just uniformly spaced out, like on a grid, but that doesn't need to be the case. This framework that we're using, BioCellian, it allows you to be off-grid, so you could be in any XYZ location. And so that would be also interesting later to look at what happens when cells can move or they can have different distances from each other. But for now, we just wanted to start simple. And each cell has a signal that comes into it. This is a molecule, a diffusible molecule. It has a receptor node that senses the concentration of the molecule. It transmits that into an internal network, a Boolean network. And then there is a signaling node that also sends out another diffusible molecule. Right now, we just have one diffusible molecule, but later we can also look at different, more diffusible molecules, more than one. And you already have seen this, Boolean networks. Cells inside are basically synchronous Boolean networks. Okay, now, in this case, we are not going to use stochastic dynamics. We are not going to create an ergodic Markov chain and look at steady state distributions. We're actually going to let this thing go to attractors, the way Stuart Kaufman talked about this morning. So if you wait long enough, it will get locked into some sort of a periodic, repetitive pattern of activities, and that's an attractor, right? So it's a deterministic system. It must eventually go to an attractor. And so we will wait for the transient behavior to disappear. And once the system goes into a stable sort of attractor, then we will analyze what happens. So this is to say that we have a burn-in period. We basically throw away a bunch of time points at the beginning and wait till the system stabilizes. Now, how does this cell communicate by diffusible molecules? Well, I already mentioned that it secretes these molecules. And of course, you have to solve partial differential equations if you want to do this in 3D. Or you can use a steady state instantaneous decay assumption, which I'll show you. But basically, you have to assume that there is some secretion rate of a molecule. And if the signaling node is 0, then it's off, and you have some basal secretion rate. It's still secreting, but with some basal rate. But if the signaling node is on, then it's secreting with some alpha, some multiple of the basal secretion rate. So it's greater. It increases its secretion rate. So that's one parameter that you can control. The concentration at any given point, radius r, from the cell assumes an instantaneous exponential decay, a diffusion equation, shown here on the bottom. But essentially, it's a function of this parameter eta, which is the secretion rate, the radius, or the distance from the cell, and which is, the effective it's also in this equation. So basically, it's the diffusivity of the molecule, how far that molecule can travel. And then finally, there's another parameter, K, which is the uptake threshold or the activation threshold. What does the concentration have to be in order for the receptor node to turn on, to become activated, right? It's some threshold. So K is that threshold. And you sum all of the concentrations from the other cells at any given point to read the concentration. So all cells communicate. That's the difference. It's just your nearest neighbor with east, west, north, and south. Now, this cell can communicate with faraway cells just because of diffusion. Okay? But it's not specific. So that's the difference. It's not specific wiring. So our two parameters that I'm going to show you in the next several slides are going to be lambda and K. Those are the, let's call them the communication parameters. Lambda, again, is the diffusivity, how far the molecule travels, and K is the activation threshold. So they're related, of course. They're sort of on two sides of the same coin, but they are independent parameters. So in order for us to even get a sense of what happens, we wanted to start with something super simple, and the cell just had a receptor node and a signaling node, nothing else, no network inside. This is just to get started. And this setup, we wanted to ask, well, what happens if we vary these parameters, lambda and K? What happens even in the simplest of cases when there is just a receptor and a signal node and nothing else? Well, we found that as a function of K and as a function of lambda, we have 6 different regions of behavior. And they're labeled with Roman numerals 1 through 6. Okay, sorry. So in region 1 and in region 6, cells are getting input only from other cells. Okay, so here lambda is so high and here K is so high that here cells are either always on and here they're always off. In this little region, number 4, cells are only listening to themselves. They cannot hear what anybody else is saying because, you know, they produce the molecule and they can sense it themselves, but the other cells are too far away and the parameter lambda is too low, so they cannot see what the other cells are doing. So here, the cells are basically kind of like autonomous. And the interesting regions are right here, 2, 3, and 5. So here, there is sort of, there is a mix of signals that the cell is getting either from itself or from other cells. Here, it's really primarily getting signals from other cells, but it's mainly getting turned on or getting turned off. But in these narrow regions, 2 and 5, that's the most interesting behavior. That's where they're getting signals from both other neighbors and other cells and from themselves. So we decided to extend this to much larger K and much larger lambda. So you can see that this whole graph fits inside this little box right over here. So we wanted to see, well, what does this look like? It looks like this. And you can see that indeed these regions 2 and 5 are very, very narrow. It's a very narrow space where interesting behavior happens. So we created, here are some results. We created a tissue with 16 by 16 by 16 grid with periodic boundary conditions. Every cell has a random Boolean network with 10 genes inside. So if you put everything together, there are like 40,000 genes or nodes in this whole thing. We give them random initial conditions. And then we start sampling different lambda and K values. Again, these are the communication parameters, so they tell us how much communication happens between the cells in these different regions. Then we simulate, we wait until we get steady state behavior or attractors, and then we analyze. We look for cell attractors, tissue composition, and spatial organization. And we do everything 100 times just to get good statistics. So, what we see now about possible cellular behaviors. On the x-axis, you have the number of cellular attractors. Since this thing is deterministic, right? There's no noise here. I can actually count how many attractors each of these cells exhibits and do empirically, just by counting. This is a histogram of the number of attractors, number of tissues possessing that many attractors. So how many tissues have 2 attractors? How many tissues have cells with 4 attractors, 10 attractors? Now, these simulations are in region 1, 6, and 4. That's right here, 1, 6, and 4. These are the boring regions. You can see there are not that many attractors, 4, 6, 10. But if you go into these more interesting regions right here, the blue ones, now you get lots more attractors. This is a log scale. So this is many, many more attractors, like 100 attractors or more per cell. And so you get much more, new behavior of cells when you allow them to communicate in this way. You can also ask, well, how many cells switch their behavior? So they used to have one behavior, but now you allow them to communicate a little bit more, and now their behavior switches. So the way we can do this is we can ask, well, relative to some region, like in this case, region 4, which is over here. Remember, this was the boring region where cells only can talk to themselves. Relative to that region, if I drag away, if I move K or I move lambda or both, right? How many cells will start to have new attractors? That's this measure, and that's shown in this graph. So you can see that there's, again, a strip over here where you get new attractors. But if you go away from that, you don't get any new behaviors. Okay, and this is the same thing relative to region 1. That's why this is black. Obviously, relative to itself, there's no change. That's why it's 0. But as you move away from it, you see a really rapid change. Many more cellular attractors start to appear. Now we can also ask how many cellular attractors can coexist in the same tissue at the same time. And here's the result. Again, there is this strip right here where you have most cellular attractors. Away from that, you have pretty boring behavior, kind of homogeneous behavior. We can also look at another measure. By the way, this stuff is unpublished. The first stuff was published, but this is still a work in progress. So another measure is Shannon entropy. We can simply look at the cell types as labels. We can just call them cell type A, cell type B, cell type C, empirically, right? Because we know that they're unique. We can count the number of states that each cell is in. We can say, this is attractor A, this one is an attractor B. They're just deterministic systems. So now we can look at the probabilities of each of these attractors, or let's call them cell types, and we can compute the Shannon entropy of that probability distribution to basically ask, well, how diverse is it? And again, here you see a similar result. Maybe this is already starting to look boring because all these plots look similar. You have this bright area here where you have highest Shannon entropy, just in that narrow range. I'm going to skip this. And this is actually my final slide. This has to do with spatial diversity. So if you look spatially, how diverse are these cell types or these behaviors spatially? So for example, if you had something like this, where every other cell was, blue, purple, blue, purple, blue, purple, then that would not be very diverse. Whereas something like this would be more diverse, because some areas would have more purple cells, some areas would have less purple cells, and fewer, and so on. So the way that you would do this, and I think this is something that's done in ecology as well, is to look at subsets of the whole simulation domain. So this is 16 by 16 by 16. So you look at like 2 by 2 by 2. And you ask, well, what's the deviation in that little 2 by 2 by 2 block relative to the rest of the domain? And you keep repeating that with different, so you keep taking little 2 by 2 by 2 chunks, and you look at the dispersion, and then you compute the mean absolute deviation from the mean, or from the median, I don't remember actually, of this dispersion. And again, you see the same result. that the maximum spatial diversity happens in those narrow regimes of K and lambda. To conclude, in the first part of my talk, we showed that even in a relatively simple model system, 2D model system of a tissue with isogenic cells, you can have a natural emergence of differentiation. And we see that in our simulations. We also see that the highest diversification occurs near the critical regime when the Lyapunov exponent is 0. And this is consistent with a lot of a big body of work showing that greatest diversity occurs near the critical regime and other sort of optimal properties of systems. Now, we also showed that you can tune communication parameters to have very distinct tissue behaviors or overall system behaviors. So you can imagine that you could use this even in an engineering way, where you would create synthetic systems or perhaps evolving systems or mobile populations, where you could somehow take advantage of that idea of tuning the communication strength and type, perhaps, in order to elicit global system changes. We also saw that stable cellular behaviors can be lost or gained purely by changing communication. So if you take a cell by itself, it has a certain behaviors. But if you allow it to receive some inputs from other cells, it can gain new behaviors, or it can lose previously existing behaviors. And we also saw that changes to tissue composition and cell behavior plateaus as the range of communication increases. So in other words, if you look at these graphs, basically it plateaus. You have diminishing returns. Eventually, you don't get more diversification no matter what you do. Even if you try to increase these communication parameters, nothing is going to happen. But the other thing is that there is a very narrow range of communication in which this diversity and spatial organization can be maximized. So it's not a big range. It's a very narrow range within which you can get this diversification. So it's still an open question, and it's an interesting question. What features of these systems are actually affording this, this behavior of diversification? Is it simply that they are critical? Or are there other properties of the systems that afford that behavior? Okay, so that's it. Thank you for your attention. Thank you.
Thank you.
Thank you for this talk. I wanted to know, very simplified model where cells could only have 2 attractors, let's say attractor 1 and attractor 2, so only 2 possible outputs. Would you recover a simple Ising model with an external magnetic field for the... You see, I was wondering because
yes.
You have the nearest neighbor communication, and then you have the mean field that, you know, and if the cells can have only 2 attractors, then you should be able to recover the known Ising model, you know, results.
Yes, that's a very interesting thought. I know that these checkerboard patterns have been seen in like anti-ferromagnetic, systems. So, yes.
There should be a bridge somewhere. Yes.
Yes,
okay.
Very interesting idea. Thank you for that.
Another question?
Thanks for your talk. It was very, very interesting.
Thank you.
I have 2 questions. One is, have you considered checking this against the biology? Because when you are...
Have I considered what?
Checking this against the biology, because when you are tuning the parameters that you have, it will be equivalent to saying that the receptors have a threshold of activation, not only of affinity, but of activation. And you can, for example, try cytokine concentrations, see if there is a narrow range of concentrations that properties of a tissue. So that could be interesting to see if your system is predicting the range of response of the natural receptors. And the second one is the model you propose as multicellularity is a model where all the cells have the same genotype, so you're supposing that you are coming from only one cell that is expanding and then differentiating.
Yes.
That's usually what we suppose happens with animals, and kind of with plants. But for example, Myxobacterium, they have aggregation. So you have different bacteria that flock, literally. They're very pretty. And then they have different genotypes and different networks, and they create tissues for sporulation. So have you considered checking against that?
Yeah, great. Thank you for your questions. Indeed. No. So the short answer is we have not checked. This is not supported by any experimental work, but I agree that it would be great to figure out a tractable system, like a tissue on a chip or organ on a chip or some kind of a multicellular system where you can tell spatially, maybe in 3D, maybe in 2D if it's like epithelial cells, but to then give them some, growth factors or cytokines, and to do what you suggest, to actually see whether their behaviors will change. But I suppose that would require some kind of pharmacological inhibition of receptors, perhaps, where you can simulate the effect of tuning the lambda and K parameter. Like, oh, right, exactly. Exactly, right. So by competitive binding, right, use antibodies. So that would be one. And also, yes, you're absolutely right with your second question with the bacteria that, you even have horizontal gene transfer. So, which is another level of complication. So not only do they flock and they have sort of a community, and they also have a horizontal gene transfer. They can kind of exchange pieces of their DNA. And so, yes, this is another level of complexity. You're absolutely right that the assumption here has been from an isogenic, cell, and that's diversified. But yeah, it's an assumption. Thank you for your question.
So, any of all this sort of work, even from Kaufman, how often do you put selection in? I mean, it sometimes seems this is like the grammatical element, but the semantic element, meaning what a cell being particularly useful or not useful, has to be basically governed by natural selection. You can have many or you can have few, but it's your environment that has to say that's a good cell or something that's useful to me.
Yeah, it's a good point. We did not look at evolution here at all. Or evolvability of such networks, and that would be an interesting thing to look at. I know that actually Max has looked at evolvability with colleagues, and that is an interesting an important property to understand. And so, yeah, I think that would be next steps, the follow-up work of this. Yes, thank you for the suggestion.
Another question. Well, let me ask you this, Ilya. You mentioned that this diversity emerges mainly around the criticality, the critical point. And then you showed us these stripes, like the bright stripes in your. Would you say that these stripes are also critical, or are they somehow related to the other criticality?
That's a very good question. In the 3D simulations, we did not focus so much on the criticality, but we did start with networks that were nominally assumed to be critical because we wanted to see the most interesting behavior. So we didn't start with really boring networks where we know they're going to be ordered or highly chaotic. So yeah, so I should have mentioned it. I failed to mention that all of those simulations in 3D were with critical networks already. And we just wanted to see the effect of lambda and K, the communication parameters on the behavior. But if we started with ordered networks, I think that everything would look boring. If you were deeply your K and lambda parameters wouldn't make any difference. Basically, or not much.
Did you try chaotic networks?
Yes, I think yes. My student tried both ordered and chaotic networks, and we got, I think, generally pretty boring behavior, which is why we settled on critical networks for this investigation.
Okay, I get your word.
Well, it depends on how chaotic, right? Yes. How far away you go.
How far away?
Yeah.
Okay. Are there more questions? Yes.
In this case, how do you define the criticality of these networks? Or how do you select for a network for having this level of criticality, or how do you measure this?
In this case, we construct networks with K and P such that it is equal, so that 2KP 1 minus P equals 1, that equation I showed. So that's actually how we construct. Now, we can go back and verify it by computing the average sensitivity and the Lyapunov exponent to actually see whether our given network is even close to being critical. If it's not, we can throw it away and generate a new one. That's how we do it, actually. Because we can check a given network, we can check its Lyapunov exponent. We can compute it explicitly. So we just generate networks until we're happy that we get Lyapunov exponent 0. It's not the most efficient way of doing it.
Another question? Well, if not,
Thank you.
Let's thank Ilya again, please.
Thank you very much. Thank you.
Don't go, this talk is going to be very good. Okay. Hello. Hello, everyone. No, aquí está él, mira. Ven, firma. Firma esto. Es la autorización de que esto salga en Facebook. y en YouTube. Ah, no, claro, claro. Ahí está.
Derecha, izquierda, y, bueno, para adelantar, para atrasar y el apuntador.
Sí, pero, ok. No, no, no. It's already here. Hello. Next speaker. Yes. Oh, yes. Yeah. Well, this is going to be very strange. I have never seen this before. It is a great pleasure to introduce myself. I don't know. Hey guys, we have just started, please. No, really, it's we are three organizers and Murphy, okay, the guy of the Murphy's Law, if you remember. So we are three organizers and Murphy. Murphy has been with us since this morning. So supposedly Saul Witzel was going to give this presentation. Saul Witzel is a collaborator. He is here. He doesn't feel well as to present the talk, so I'm going to give the talk. That's all we're supposed to give. It is exactly the same thing, but this work is Saul's PhD research, and it's my great pleasure to present his work and to introduce myself again. I would like to thank the organizers. No, we are the three organizers, but somehow, you know, they are attending some other issues. So let's start. Saul, project to Saul, he's a physicist. He learned a little bit about computer programming, but we never thought that he was going to take this project to the level that I am going to show you. Then there is Santiago Sandoval, who is another organizer of this conference, and he had to go because he had an appointment with the doctor. And then Alejandro Frank, who is the director of the center, is another collaborator, and myself. So let me start out with this phrase. This is John Maynard Smith. This is one of the greatest evolutionary biologists in the 20th century. And in this book, The Origins of Life, he said that Darwin's theory of evolution predicts that organisms have to become more adapted to their environments, but it never explains why they should become more complex. From this perspective, life on Earth should have never gone beyond bacteria. Okay, why? Because bacteria, they can adapt to any and every environment that you can imagine, right? So there is no reason from the point of view of Darwin's theory of evolution for complex organisms to have emerged. I mean, bacteria is already enough. They can adapt to very humid conditions, to very dry conditions, to very hot conditions, very cold conditions. I mean, you can think of any environment, even radioactive conditions, they can adapt to that. So what's the reason in evolution, well, from an evolutionary perspective, what's the reason? What's the need for complex organisms, multicellular organisms? And this is one of the questions we would like to address today. Keep this problem in mind. And Ilya was talking about multicellularity. And it happens that multicellular organisms, multicellularity now has been reformulated
because we are multicellular organisms in many different ways. We have different cells, different human cells, but we also have different bacterial cells. And these bacterial cells are an important part of our metabolism. So it is estimated that the human body has about 10 to the 13 bacterial cells. This is more or less between 1 and 10 bacterial cells per human cell. And altogether, they have about 100 times more bacterial genes than human genes. So we're of bacteria. So we have bacteria in the guts, in the skins, everywhere. Okay. So multicellularity has reached another level. It's not that we are multicellular organisms because we have different human cell types, but because we also have a great variety of bacterial cells in our bodies, which are related to our metabolism in very complicated ways. Now, these bacteria that we are carrying on are not just there doing nothing. They actually interact with. It's not that these bacteria jump onto us and then colonize our bodies. No, we were born with them and we interact with them in many different ways. Bacteria, they regulate fat accumulation in our bodies, formation of vessels. They participate in the development and training on the immune system. They participate in the biosynthesis of vitamins and amino acids, and so on and so forth. So all these bugs that live with us are an integral part of our metabolism, and we know this now. We didn't know this 15 years ago or so, but now we know that these bacteria play important and fundamental roles in the development of multicellular organisms like plants, insects, animals, and humans, of course. And here there is just to give you an idea, it has been found, I'm sorry. This should have not happened. Here it is. So in our stomach, it is estimated that there are more than 25 species of bacteria, different bacteria in our guts. There is over 1,000 different species of bacteria. On our skin, over 1,000 species. In our lungs, 600 different species, and so on and so forth. So it's not that we have just a couple of different species or a very few different species of bacteria living with us. There is a great variety. There is a huge variety. Thousands of different bacterial species living with us. So the question, we can formulate these different questions. What kind of evolutionary mechanisms are able to generate symbiotic relationships? Because these bacteria are symbionts with us. Did symbiotic interactions occur just because they are possible? I mean, throughout evolution. What are the general benefits that bacteria confer to the host during this evolution? And these two questions are related. I mean, did symbiotic interactions appear just because they are possible? Is related to the question of why the host could not simply evolve by itself, right? Why the host has incorporated so many different bacteria to perform different metabolic processes. Of course, an obvious answer is that, okay, if in evolution there is a function that is already invented, if there is a problem that is already solved, then different organisms will take advantage of that problem and just incorporate it instead of reinvent it. And then the other question, why microbial diversity is so high? Why do we need so many different bacterial species living with us? And why complex organisms are necessary? I mean, bacteria, they have already solved all these biochemical problems. What is the need to have complex organisms with lungs, different tissues, and different functions? Now, these are not just rhetoric questions. These are actually scientific questions that have generated a very crude debate between two different groups or more groups. This guy, Rosenberg, Eugene Rosenberg, he proposed the idea of the holobiont as a unit of selection in evolution. The holobiont is the host plus all its bacteria. Okay, this is the holobiont. And he says that the integration, the correlation or the interaction between different bacteria and the host is so close, is so fundamental that we cannot think of the host as an individual. Or, if you want, the concept of individual has to be reformulated in evolution because it turns out that individuals are not longer individuals. They are like this guy here, okay? We are a bag of different bugs, literally, including our own cells. So if we are going to formulate a new theory of evolution, we have to consider the individual not as an individual, but as an ecosystem, right? And this ecosystem is the holobiont. And this holobiont is a unit of selection in evolution, okay? So he publishes this paper about 10 years ago, and he says, I don't know if you can see it, but I can read it. We present here the hologenome theory of evolution, which considers the holobiont, the animal or plant, with all of its associated microorganisms, as a unit of selection in evolution. The hologenome is defined as the sum of all the genetic information of the host and its microbiota. And this has to be considered as a unit of selection in evolution. Of course, 1 year after that, I know here's the zoom in of this previous slide, so you can read it now. 1 year after that, a competing group published this paper saying the whole genome concept had hollow. And this was a critique, a very strong critique to the concept of the whole genome as a unit of selection and evolution. The main argument is that Darwinian theory already considers the evolution of ecosystems. So we do not have to adapt the Darwin's theory of evolution because it can already deal perfectly well with ecosystems. And then they reply back saying, well, yes, but getting the whole genome concept right. Okay, this is like, you didn't understand what I said. Now, please listen, pay attention. So what they say is, yeah, but in an even in the Darwinian theory of evolution, an ecosystem is composed of individuals. And now, the individual is a whole ecosystem. So there is this big debate, which means that the questions that I was asking before are not trivial. The answers are not obvious. It is not like, oh yeah, of course we have very specific examples in which the bacteria help in a very specific task to the host, okay, like digesting fat or digesting cellulose or processing whatever other molecule. So there are many, many examples, but the general questions as to why do we need so many bacteria, how these symbiotic interactions emerged in evolution, those are not trivial. Here there is the answer of the detractors, holes in the hologenome, why host microsymbiosis are not holobionts, and so on and so on. So there is this recent debate. You can have fun in the papers. And then this guy, Bordenstein, principles of holobiont, 10 principles of holobiont. So these are the 10 rules that holobionts must follow. So yet another one. Okay. The main two criticisms after reading all these papers, my conclusion and our group's conclusion is that the main two criticisms to the hologenome concept are this. Evolutionary conflict, this is one. microbes and the host may have different interests, so they cannot always co-evolve. Actually, most of the time they cannot co-evolve. They cannot live together because they have competing interests, and this is known as the evolutionary conflict. And the other is the lack of a core microbiome. I mean, the microbiome that I have is different from the microbiome that he has, from the microbiome that she has. So there is not a core microbiome. So it is not possible as a species to evolve if we have this diversity in the composition of the individual microbiomes that each one has. So the lack of a core microbiome, the microbiota is too diverse. So it cannot be possible. These are the two main criticisms in our opinion. So we want to address this problem from a systems biology perspective, okay? In one of these papers, the authors, Rosenberg, said, okay, this is just blah, 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 but to be honest, there is evidence in one direction, there is evidence in the other direction, but what we need is mathematical models, mathematical and computational models that can prove or disprove the hypothesis of the holobiont as a unit of selection in evolution. So we're trying to do that, and we choose to work with Kaufman networks. Kaufman networks, we already know now after Ilya's talk and after Stu's talk, that these are models of gene regulatory interactions that are able to reproduce real gene expression patterns. So when they were proposed 4 years ago, they were not very well accepted in the biological community, but now everybody, this is like the standard model. Okay, everybody, when you have a genetic network or even a metabolic network, the first thing you think of is to use a Kaufman network to represent the dynamics of that system. So this is what we did. We use the Kaufman model. And this is going to be important at the end of the talk, not for what follows, but just at the end. This is the phase diagram of the perturbation in this Kaufman network. So if I perturb a gene in these networks, let's say that one gene is activated and then I artificially turn this gene off, then this perturbation may propagate to the other genes that are regulated by this one, and so on. And this propagation can then propagate across the entire network. And this is the size of the avalanche of the perturbation. So as a function of this sensitivity that Ilya was mentioning before, the size of the avalanche, the final size of the avalanche follows this phase transition curve. If the sensitivity is less than 1, then the perturbation disappears. And if the sensitivity is larger than 1, then the perturbation propagates to, this is for an infinite system. I mean, computed analytically for an infinite network. So here it means that the avalanche already propagated to an infinite fraction of the system, a considerable fraction of the system. And this is the critical point, when the sensitivity is 1. And let me show you graphically how this phase transition occurs. Where is the? Here. So this is a Kaufman net. These are the genes. I place them on a square lattice just for visualization purposes, but this is a random network, okay? This is not a square grid, it's random network. So the topology, the connections, we cannot see them, but the connections are random, just for visualization purposes, and also to convince the biologists, because they like to arrange genes in microarrays like this. So this looks like a microarray, okay? And this is a network, but the connections there are random. And then let me change this. This is the order regime. See? Time is going on, but the network is frozen. Okay. Now, if I perturb some of the genes, let's say some of the genes that are on, I put them off, and vice versa. These are the red. So this is new perturbation. Okay. So this is the perturbation. I forced some of the genes to change its value. And now see how this perturbation propagates in the order regime. Ta-da! It disappears, as Ilya said. Okay. Now let me put this network in the chaotic regime. Now, a new perturbation. Okay. Actually, these are too many genes that are being perturbed. Just let me perturb less genes. This is the size of the perturbation. See, I can control everything here in my program. That's it. Yeah, very few genes are perturbed. And then the perturbation propagates to the entire system. So clearly, genetic networks of real organisms cannot operate in this dynamical regime because even the smallest of the perturbations could change the dynamical behavior of the entire system, right?
What is XT?
XT is the fraction of red squares.
Okay, thank you.
There. We need the mic because it has been recorded and transmitted to other places. And without the mic, the questions are not listened, are not broadcast. Okay, so XT is the fraction of the genes that are being perturbed. This is, and you see, nearly half of the genes are being perturbed. This is like reinventing the organism with even the smallest of the perturbations, whereas in the order regime, the network doesn't respond to any perturbation. It cannot evolve. And then typically what happens in the critical regimes, these are critical networks, and this is a typical example of what happens, that the perturbation neither disappears nor increases, doesn't propagate to the entire system, but also disappears. But then it remains confined within a subset of the genes. So this is why Stu postulated many years ago that living organisms should operate in the critical regime, because this is the typical response of the network under perturbations. And this is this particular point here, okay? When the networks operate here in the critical phase, they behave like this under perturbations. Now, this is going to be important by the end of the talk, not right now, because we are not interested now here. What? My mark. There. What we're going to do is to train this network to perform a particular task. This is what we want to do. Imagine that this is the genetic network of organisms. Let's call it the host, okay? And we are going to train this network to do something. Now, network training is a very old area in computer sciences, and we can train networks to recognize pictures, to recognize sounds, to do mathematical operations. And usually, computer scientists use neural networks. Well, now we're going to use Kaufman networks. Why? Because these are representative of how they interact. Okay? So what we're going to do is to choose a subset of the nodes in this network. These are the genes. And we are going to extract a signal from these genes. In this particular case, the signal is just the simple sum of the states or the values of these genes. So let's imagine that there is suddenly the environment changes. The organism has to cope with this change, has to develop some new functions, cope with this change. But of course, it's not the entire genetic network that is going to change. It's just a subset of genes. For instance, when you apply shock to yeast, it is a subnetwork of about 15 genes that responds to this heat shock. Okay, when you compress E. coli, there is a subnetwork that responds to this perturbation, and it consists about of 20 genes. So it's not the entire genetic network that responds to the environmental change. It's a subset of genes, and then we are going to extract a signal from this subset of genes. And we want this signal to behave in a predefined way. This is the target function that you can see here. Let's say that this is the appropriate response of the network to the external change. This is how the network should behave, or these genes should behave in this way. However, after the change has occurred, of course, the network doesn't have this phenotype, so it behaves in a random way, especially because these networks were constructed randomly, so they don't have this output, the desired output. They behave like that. So the question is whether we can train the Kaufman network to have this signal instead of this one. And the answer is yes, we can do it. Okay, we have to define our fitness function, and the fitness function is the average square error between the target signal and the actual signal. Okay, so this is the function the network has to perform, and this is what the network is actually doing. So we measure the mean square error between these two functions, and we minimize this error. So we introduce these networks into an evolutionary algorithm, a very traditional algorithm, so that after some generations, what we are going to do, we are going to mutate this network. We are going to rewire some of the input connections, some of the output connections. We are going to change the Boolean functions, right? And in each generation, because we have a population of networks, in each generation, we are going to choose the best networks, and so on. And eventually, you see, the signal actually transforms into the target function. This is the evolutionary process. It's nothing complicated. It's a very traditional thing. We were not creative in this point. So this is the population. The error is colored according to this color code. So networks with a large error are not well adapted. They are painted in red. Networks that are very well adapted, they are painted in white. So we start with the population of red networks or reddish networks because none of them can perform the function. We order those networks and we select the best ones, and then we repopulate. And then we mutate. So this is the evolutionary process. Then here we mutate the networks and repeat the process. And eventually, the error will decrease in the population. Okay, so ordering, selection, and repopulation is one generation. And these are the results. So this is the host network. I want to abuse a little bit of the language, and I want to call this the human network, okay? This is the human network. It's just to have a concrete idea in mind. The white curve here is the target function. The target function doesn't have to be as nice as before. It can be an arbitrary function. And then the green line is the actual signal, what the network is doing, okay? And then here you go. So you see that we mutate these networks, And throughout generations, the network signal actually approaches the target function. And in the little inset that you have here on the left corner, you can see the error between the signal and the target function, and you can see how this error decreases in time across generations. Okay? Of course, this is an evolutionary algorithm. At each generation, selecting the best network, so we expect the error to decrease. So up to here, there is nothing really surprising. I am just showing you that these networks can be trained, okay? And that the signal, the output signal, can actually approach very well the target function after some generations. This is the average error in the population. So we start with networks with a very large error. And we see that after generations, the error decreases until it reaches what we call the adaptation threshold. We set this adaptation threshold at 1. And this means that when the network reaches this error, there is only one node in the signal that is different. So the signal is very well appropriated almost at every point, except probably at 1. Okay? And we could say that once the population reaches this threshold, it is well adapted. Yep.
I had a question about your error bars. So it's the previous slides. I wanted to know why the error bars were not like disappearing. If you find the perfect target at some point, they should just disappear.
No, no, they do. Look, compare the.
No, but they seem to be steady at some point. I was wondering if there was a degree of freedom there.
No, no. Well, I don't know, to be honest.
You see, compared to the average, that decreases drastically.
Yes.
Then the error bars, they stay somewhat at some point at the same value. It seems more painful, the convergence there.
Yeah.
Yes.
I don't know,
okay.
To be honest, I don't know.
So if you go back to the... Yeah, so is that kind of a representation of the population fitness? Because you got mutation in there and things, right?
Yeah.
So the thing is, you're not going to have a completely converged population. So you're always going to have probably different networks that have within there that are around. It's a little bit like a quasi-species or something like that. You'll probably have a little population of networks, and some are going to have worse performance, whatever respect to the ideal one. Otherwise, it should be the case that we should converge to something perfect. Right?
Yeah, thank you, Chris. Yeah, at every generation, we still have mutations and this variability in the population. And probably what we are seeing here is the steady state distribution. I think so, yes. I would say so. I am not sure, but it seems reasonable. The question is, okay, we have seen how the human network can adapt to a particular task. Now, what happens if we let this human network interact with a microbial network? And what is the difference between the human network and the microbial network is the mutation rate. Why? Because bacteria, they can generate mutants at a rate that is at least 10 times larger than human cells, okay? So these networks are nearly equal, the same size, number of nodes, same connectivity. What changes from the human to the bacterial cell essentially is the mutation rate. And one important point is to test the evolutionary conflict criticism is to start with bacterial networks that are already trained to perform specific tasks. So you can see here that the bacterial network already performs very well its function. Okay, so this bacterial network is already trained, whereas the human network is not trained at the beginning of the evolutionary process. So it's like trying to simulate the fact that from the environment, all of a sudden, a bacterial colony jumps into my body and they start interacting. The bacteria already knew how to do something, okay? And see if they can help the host in adapting to the task and preserving its own functionality. And yeah, they can do it. See? They're helping the host is adapting to his task, and at the same time, the bacteria, the bacterium remains adapted to its own task, okay? And you can see here now the error, pay attention to this little corner here. I'm going to point it. Before, to cross the adaptation threshold, it took about 400 generations, the host network. Now it is less than 100 generations. I mean, interaction with this bacterial network speeds up the adaptation of the host very considerably.
But now you have to explain why that's preferred to just having the host itself speed up its own mutation rate.
Yeah, that's a whole issue. That's a whole issue. I mean, the mutation rate is a parameter that is also under selective pressure, okay? And Chris, yeah. The thing is that the mutation rate cannot increase very widely when you have complex behavior, as you will see later. This is the final part of my talk. Okay, so you're jumping ahead. If you give me a few minutes, I will explain you that. Why the host? And yeah, ultimately, why, yeah. Through the Boolean functions. This gene has some regulators in the Boolean function, and then you add a new regulator, so you have to expand the Boolean function to accept a new input. This is what we do. Oh yeah, of course, yeah. Zebrafish. Over 200 genes of zebrafish are regulated by its microbiota. Yes, that is known.
Max, when you expand the Boolean function to accept another input, how do you expand that function? I know it's a technical question.
If the input is off, if the new input is off, we just copy the old Boolean function.
Okay.
When the input is on, we reassign randomly the new table.
Thank you.
Yeah. How the bacteria helps the human to perform a different task than the bacteria is actually performing, because the two functions are different, right?
Yeah,
so
these two functions are different.
You have something which is completely different that helps to do something which is also completely new.
Completely new, yeah. Yes,
okay.
That's it.
And you can choose the function as you wish.
Yeah, this is an arbitrary function. Absolutely.
So if you have some diversity here in these functions, then it will work. That's more or less the idea.
That's right. Yeah. Now, the mic, please.
Just to clarify, from the green network perspective, the bacteria are seen like perturbation, external perturbation.
That's
right. Okay. So that's,
yes.
They are not because they are not mutation. It's not like increasing mutation rate. They are just the green dots are sometimes perturbed by the red network. Okay.
Yes.
And vice versa.
And vice versa. Yeah. Well, now here we have the results. This is the error when the host network is evolving by itself, and this is the error when it is evolving, interacting with the bacterial network. And you can see that it reaches the adaptation adaptation threshold much faster than when it evolves by itself. Not only that, but this is the number of accumulated mutations throughout generations in the host without the bacterial network and with the bacterial network. And you see that the number of accumulated mutations in this particular example almost halves, okay? So it's not only that the host network adapts faster, adapts better, because you see here that the error is much slower. You may consider that this difference is not that big, but we are reaching the threshold, the ultimate capability of the network to perform the function. So here, this difference is huge because this is almost a perfect adaptation, okay, to the task. So it's not only that, but also the human network has to mutate less, considerably less, to perform that task. So it seems that the bacterial network is doing the job, okay? It is the bacterial network that is assuming, receiving all the mutations and actually working for the host. Now, what happens if, after they have co-evolved, we disconnect them, okay? What we expected, and let me tell you this, what we expected was that, okay, the host network was trained in the presence of the bacterial network, so we didn't expect the host to perform well after all the connections with the bacterial network were removed. But the bacterial network was already well trained before it started to interact with the human network, okay? So we expected that after this connection, the bacterial network could keep doing its own task. But this didn't happen, okay? Well, so after they have co-evolved, we disconnect them. This is the initial error at generation 0. As you can see here, the host network has a large error because it is not trained. The bacterial network has a small error under the adaptability threshold. After 500 generations of coevolution, both errors are considerably smaller, below the adaptation threshold, and then we disconnect those networks. And this is what happens. This is typically what happens, that the error in both networks, in both of them, reach levels or values that correspond to completely untrained networks. So they became symbionts throughout this evolution, and now none of them can perform their respective tasks without the help of the other. And this is a very well-known result. This is evolution, and this is disconnection, and this is a very well-known result that when you decrease the diversity of your microbiota, for instance, with antibiotics, then all these diseases known as dysbiosis emerge, okay? So you kill some of the bugs that you have in your guts, in your stomach, and you start feeling sick because you need them. And also, many of these bugs cannot be cultivated outside of the host. This is why the microbiota was not discovered a long time ago. It was discovered just recently because most of the bacteria that constitute the microbiota cannot be cultivated in isolation. Now, of course, if one bacterial network helps the host to adapt faster and with less mutations, well, the hypothesis is that the more bacterial networks we use, the better the adaptation of the host. And this would resemble the great diversity of the holobiont, right? In the microbiota of the holobiont. But this was not the case. Actually, we could see in our simulations the emergence of evolutionary conflict when many bacterial networks were added to the microbiota. See, this green line is the host evolving with 0 microbial networks, just evolving by itself. This is the green line here. Then we add 1 microbial network, is this dashed line. down here. But then happens when we are 2, 4, 6, 8, and 10 microbial networks. The adaptation of the host is worse and worse, okay? Oh. Sorry. So it seems that adding diversity to the microbiota actually worsens the adaptation of the host. And this is evolutionary conflict. And Saul says, now let's imagine that we are fixing our car. We want to change a flat tire. And then if there are 2 people trying to do it, they can cooperate and do it very efficiently. But if there are 10 people trying to change this flat tire, they are going to obstruct each other. And they're not going to be able to actually be efficient and do the job. And this is exactly what is happening here. When we add 10 microbial networks, you see that within the computing time, the host is not able to cross the adaptability threshold. Okay, it's like all these bacterial networks are trying to change the flat tire, and then they obstruct each other. But this is because in all these simulations, the host network. Yeah, I'm sorry, I'm a little bit short of time. Can we just wait a little bit, or if it is very quick? No, I'm not supposing that. Why? Because we know that the whole genome actually is structured in modules. Okay, so you can consider that this is a module, a small module. Yeah. So in all these simulations, the host network has to perform only one task, and then you have many bacterial networks trying to help it to perform this single task. But as I mentioned in the introduction, there are many different tasks that multicellular organisms perform: the development of vessels, the digestion of food, blah, blah, blah. So we have to upgrade to a multitasking network. So now the host network is going to perform many different tasks depending on the initial condition. So if we start with a given initial condition, the network is going to perform this task. If we start with a different initial condition, and so on. Okay, so we want to train this network to perform multiple tasks. And in this case, there are 2 interaction schemes with the bacterial network. So this is the host. These ellipses represent the different functions that the network has to learn. And then these purple circles represent the bacterial network. And in this non-specialized interaction scheme, all these bacterial networks can interact among them, and they can help the host network to perform any of its tasks. Okay, this is the non-specialized scheme, which is not very realistic. You just implemented it to see what happens. In the specialized interaction scheme, this is the host network. These are the different tasks it has to perform. But now the microbiota is divided into different independent niches, okay, like this. So that the microbial networks that belong to one niche are going to participate in the adaptation of the host to only one specific task. The microbial networks in a different niche participate to a different task, and so on. And of course, microbial networks within the same niche, they can interact, but they cannot interact if they belong to different niches. And this resembles this fact that we have bacteria, very specific bacteria in our nose, different from the ones that are in our skin, different from the ones that are in our stomach. So it's not that all the bacteria that we have are mixed together in this homogeneous soup. They are separated in different niches. So let's see what happens. For the non-specialized interaction scheme, this is the host network evolving by itself. And then we add 1, 2, 3, 4, 10 bacterial networks, and nothing happens. There is not much difference, right? They improve the adaptation of the host, but it doesn't matter how many bacterial networks you add. the adaptation is no longer improved. And this is the specialized interaction scheme. Look at this. This is the host network bytes, and then we add 1, 2, 4, 6, 10. And the more bacterial networks we add, the better the adaptation of the host when we separate these bacterial networks into different niches. And look at this. This is the host. These are the different tasks. These are the different niches. And this is the error, okay? And this is a host that has to perform 10 different tasks, interacting with 25 different bacterial networks. These are 25 separated into these 10 different niches. And this number is the average error of the host over all the tasks it has to perform. And then see that throughout evolution, all these 25 different networks actually help the host to perform the different tasks. There is no evolutionary conflict here. On the contrary. And at the end, everybody in this holobiont is very well adapted to its tasks. The bacterial networks are well adapted, and the holobiont and the host well adapted to all of its tasks. And you see that this looks like a flower, and the different change color at different rates because the adaptation to the different tasks occurs at different rates, which is fine. I mean, we do not have to adapt at the same time with the same rate to all the different possible tasks that we have to perform. And using 25 different networks, this holobiont reach this error, which is 0.41 on average. Well, this is a population, okay? So this is the average in the population. And actually, what? We did that with 100 bacterial networks. So this is the holobiont, this is the initial error. Everything larger than 3, we painted in red. So here the error, the initial error is 6. And there are 10 different niches, 10 bacterial networks in each niche. So altogether, we have 100 bacterial networks, which is more or less the ratio between the human genes and the genes in the microbiota. It's more or less 1 and 100. So this is what we did this simulation. And you can see these are different snapshots. And throughout time, the holobiont, again, becomes very well adapted to the different tasks. Now, the conclusions of this part, now there is the second part of the talk. No, I will just take about a few more minutes. But the conclusion so far is that interactions with the microbiota makes the host adapt faster and more efficiently to its tasks. The emergence of complexity is mandatory in evolution to perform multiple tasks. And what do I mean here by complexity? You remember that in the non-specialized case, we have an homogeneous network, an homogeneous big network in which the host interacts with all the other bacterial networks more or less in an homogeneous way. You think of this as a big homogeneous network. This is not a big homogeneous network. There is complexity in two different senses here, in two different ways. One, we have a different mutation rate, which is necessary. Let me tell you that if the mutation rates of the host and the microbial networks were the same, none of these things would happen. I mean, there is no adaptation, there is no improvement, there is nothing, okay? If the mutation rates were equal. We have to have this difference in the mutation rates, okay? And this is a dynamic, this introduces a dynamical difference. This is a difference in the dynamical behavior of the system, in the evolution of the system. And the second thing is this structural complexity. So we have dynamical complexity and structural complexity because we have to divide all these networks into different niches, and this provides the holobiont with the structural complexity. And only in this case is that the microbiota can improve the adaptation of the holobiont. So complexity, the emergence of complex organisms, is like a requirement for the adaptation of the holobiont to occur. Otherwise, it doesn't happen. So this is what I meant by the emergence of complexity is mandatory in evolution to perform multiple tasks, and host microcoevolution is possible without conflict. So these were the main criticisms against the whole genome hypothesis. With this model, we are not claiming that this is what actually happens in reality. This is, of course, a mathematical model that is inspired in on one of the main models of gene regulation, which is Kaufman network. So we're pretty confident that this is indicative of what might be happening in evolution. But from this theoretical computational point of view, we can answer many of the questions, many of the criticisms against the hologenome hypothesis and say, okay, see, what you're saying does not necessarily happen. Now, just to finish, and this is the last thing I want to mention, is that we want to train these networks to perform a more complicated task. Not these random things, these random functions. So we want to see if Kaufman networks can be trained to play music, for instance. And we know that neural networks can play music, yes. To my knowledge, nobody has trained Kaufman network to play music. So what we did, now the target function, instead of being this random signal, now it's going to be the notes across the music sheet for a melody. We have to binarize this music in the following way. For instance, this is the C chord. So we have here the piano keys. Each key represents a note in the network. And if I am playing this chord, the keys that have been played are going to have value 1, and the keys that have not been played will have value 0. And this is the way in which we binarize this chord. So, for instance, in this sequence of notes this sequence corresponds to this particular binary sequence. Now, I just changed the ordering of the notes. Now, I know that nobody reads notes downwards, okay? But this is just to make the analogy with the dynamics of the Kaufman network, for which time always is considered downwards. So we binarize this melody, and now this is the task that the network has to learn. And actually, it can learn the task very well. Here it is. I don't know if we have volume here. No. Yeah, we do. This is Kaufman network playing music, and it was trained with this algorithm. Okay. Yeah. Bacteria playing piano. This host network. Yeah, the flagella. Just let me finish a little bit. Just let me wrap up. It's a couple of... this is Asturias. This is another one. But let me skip this. It's just another example. Now, I want to finish mentioning this. There is this hypothesis that the brain operates also in a critical regime, okay? And you remember that I mentioned that the Kaufman networks, Kaufman, what happened to that? Yeah. that Kaufman networks have this phase transition, okay? So now, I mean, it's a curiosity that we can train these networks to play something, to play music, but if the creation of music somehow reflects this cognitive process, the creative process in the brain, this is the first time, the first opportunity that we have to actually measure in which phase this process is operating, okay? Because we have characterized the state space of these networks. So my dream now is to construct Kaufman networks that play Bach, that play Mozart, that play Stravinsky, that play folk music, popular music, reggaeton. And my bet, of course, is that some music will be in the chaotic region, like Schoenberg. Some other music will be in the order region, because here you really have to have the brain frozen to actually create or listen to the particular music. But most of the music we listen to will be here around the critical phase. Why? Because if you think about it, a good music is neither repetitive nor random. It's like a mixture of these two things, okay? So we have repetition, but also with variation. People, if you listen to a very repetitive music, eventually it gets very boring, okay? If you listen to random music, I play the keys of the piano randomly, it doesn't sound good, okay? You need these two, this trade-off between repetition and unpredictability, and this is exactly the trade-off that you get here when you operate close to the critical region. Well, this is a hypothesis, but with these networks and with this algorithm, we have the opportunity to actually test it, because we can train networks to play different musical styles and see in which of these different regions those networks operate. And with that, I would like to thank you for your attention. Wow, many questions. Yeah.
Wonderful talk, Max.
Thank you.
I would like to ask you two questions. First question, I'll start with the music. Are these networks in an attractor? Are they? So it was repetitive, just cyclical pattern?
Yeah, it was not an attractor. However, so it was a transitory. It was like part of the transient trajectory before you reach the attractor. But actually, when you let the network go beyond this particular part, it was trained during this period, or during this time, okay? So to perform, I don't know, 100 time steps of music. When you go beyond the 100 time steps, 101, 102, it doesn't sound like the music it was trained to play, but it sounds quite similar. It sounds like it belongs to that particular style. So it's not an attractor. It's not an attractor, but when you let the system go on beyond this particular trajectory, you get something that sounds quite nice, actually.
Fascinating. So quick question, number two. In the host bacterial interaction study, when you connect the bacterial nodes to the human nodes, I think those were shown as green in the bacterial network. Those like they light up as green. Those were the ones that were selected.
Yeah.
So I'm curious, so whether those were the ones that were actually selected by the bacterium alone to estimate the given function, the objective function. Do you see what I mean? Only some of the nodes are used in any network to estimate the function, right? As you showed on slide number 1, I think. A few of them are selected and they're used to estimate the function. Are those the same nodes that are selected as inputs to the host?
No, definitely no.
Okay, that's
the answer is no.
That's
actually the ones that use to extract the signal
right
may or may not regulate other nodes in the other network. Okay. But we haven't found, we look for that pattern and we found anything.
Fascinating. Thank you.
Mm-hmm.
Max, I'm here.
There is a question there.
Yes.
Yeah.
About the experiments, the failed experiments you had between the bacteria and the host genome, the one where you are accumulating bacteria more and more, and it was failing because they were like conflicting.
Yeah.
I was wondering, because I'm growing bacteria in my lab every day, and what's really important about bacteria is growth, and it's part of their evolution. And this is not included in your model. So I was wondering if we could modify it to make it successful, the first step, by just changing the relative weight of the different influence of the different bacterial genomes by having just alpha 1, alpha 2, alpha 2, and that you could also modulate, as you mutate, so that you could, meaning that one species, for instance, would be more present, and the other one will be, in a lower number, and they will have maybe a lesser influence.
No,
have you tried that? So
no, you're right. Let me be a little bit clearer. When we add different bacterial networks, these are different and they have to perform different tasks. There is no problem if you just clone the same network and make identical copies of this network, and then we use all those identical copies to train the host network. It's like having only like multiple bacterial cells of the same species. In our simulations, in that case, the training can be done. In this other case, we have different bacterial networks performing different tasks. So each bacterial network actually represents a species. It doesn't represent a single bacterium, a single organism, a single cell. So one of these networks represents a whole species. And what we are seeing is competition between species. Of course, we can assign some weight to this competition. We started with this particular, actually, we submitted this paper to the journal, and one of the questions of the reviewer was that, when do you review it, Philip? One of the questions was exactly that. What happens if you assign different weights to these interactions? And we do not know how to do it yet. Yeah, it's something that should be done. We do not know how to do it and what would be the best way to do it. But for instance, there is this cows, they cannot digest cellulose with one particular bacterial species. So this bacterial species has a lot of weight in this organism. Whereas some other bacterial species that they may have in the stomach, they don't have that much weight. So we do not know what distribution of weights. Would it be scale-free? Would it be like a Gaussian? Would it be like a Poissonian? We do not know how to do it, how to assign the weights. Of course, we can try different models, right? Yes. But the other thing is that, no, that's the answer. We have been struggling in the selection of the distribution of weights. What's the best way to do it? We will try to do that, and definitely is something that has to be done. Yeah, Chris?
So the question about the unit of selection with the hollow beyond, I mean, it seems to me you have to go one level below to evolution, whereby that flower topology emerges from something that wasn't there in the beginning, right?
Yeah.
Because I mean, that's an condition, you're actually putting it in, right? So it's a bit more, to me, like evo-devo than true, let's call it macro evolution. The other thing is that with the unit of selection, well, that's something has been around for more than a century, right? I mean, I think one of the questions there is that how do you define a unit of anything? So what do I mean by that? If you said the unit of selection is a species, I think that's a perfectly reasonable thing to say. But when a species is extinguished, it goes extinct because every single individual of that species has gone extinct, right? So the thing is that it's the same thing when a human dies. Well, what happens to his or her microbiome, right? So the thing is that presumably dies with them, right? But on the other hand, you can kill off all the microbiome through biotics or antibiotics or whatever it would be, and the person died, get very ill or whatever. So in that sense, there's also a question of the unit of selection in the sense of what happens to them from just pure life and death, right? So in other words, it's really a question of what's the scale of collectivity, right? In that sense, you can say, hey, what's a unit of selection? Well, it's a gene, but that's only defined relative to all the things that possess that particular gene, for instance, right?
Okay, well, thank you for this comment. The hologenome hypothesis doesn't say that the holobiont is the only unit of selection. It says that it is a unit of selection. And that means that the selective pressure to consider the evolution of the holobiont, you have to take into account not only the fitness of the host, but also the fitness and the interactions with the bacterial networks. So this is a good point. Probably I was not clear before. We are not saying that this is the only unit of selection, but it has to be considered as one unit of selection. And what does that mean? Well, we have to read the papers. It is because this is precisely one of the main points in the debate, and then you have this argument, this counter argument, and this other argument, and things like that. But just to clarify this point, the holobiont is not the only unit of selection. It's one unit of selection, and that means that the error, when we compute the error and minimize the error in the population, is the error of the holobiont. It's not the error of the host or separately the error of the bacterial networks. It's the error of the holobiont.
Yeah, I think the question, I think in physics, we have a nice kind of analogy of what I think of the unit of selection. It's a bound state, right? So meaning you could think, hey, well, is one gene here and another gene here in a completely different species and another gene here in a completely different species? Is that a unit of selection? And what would lead you to say yes or no? Is it like saying an electron here and a proton there and an electron here? Would I consider those in any way that it's useful to have them as an effective degree of freedom? I think that's what we're saying with a unit of selection. Does that unit represent some interesting effective degree of freedom? And what are the criteria you would use to say that's an appropriate effective degree of freedom?
Yep. Other questions? Chucho and then Francois.
Thank you. What happens if your bacteria are far from their objective function? I mean, in your model, bacterial network is close to the objective function, and the human network is far from that. And what happens if your bacteria is at least as far as the human network or even beyond?
Yeah, thank you, because that gives me the opportunity to show a movie. Yeah, I think this is the movie. In this particular case, there is no restriction on the bacterial network, so this can change freely, okay? But what we see that even if there is no restriction on the task of the bacterial network, this can change freely, right? There is no restriction here. It stabilizes very quickly. So the interaction with the host stabilizes the task of the bacterial network very quickly. So, and once we got this result, we thought about it and we sort of understand it. Now that there are these regulatory interactions that are helping the host, then the bacterial network cannot mutate freely. There are some restrictions. This training has to be maintained or sustained okay? But as you can see here, at the beginning of the simulation, it started changing very rapidly, but after a while, it becomes very stable. So this is what happens.
And the other question is, what is the meaning of a generation? Because studies, or you started your simulation at 500
generations.
In a biological point of view, what it means a generation?
Yeah, the reproduction in biological terms, the reproduction of the cell. That is a generation.
500 cell divisions.
Yes.
Yeah, okay.
Now, Francois, it is very strange to be the moderator and the
I am a bit puzzled by the almost final results you gave concerning the general network versus the specialized network. After all, the specialized network is a special case of the general network. So if you optimize the general network to death, it should just become the special. It can't possibly be worse than the special. So have you tried that? Could it work?
No. What do you mean by try to death? I mean, to wait a very long time and like in an infinite time. Well,
I mean, the general can be special, so it can't be worse than special, you see.
Probably the steady state, I don't know, I am speculating because we haven't done this, but probably the ultimate steady state is going to be the same. But the thing is that how fast you reach that steady state. And during our computing time, the homogeneous network never reaches the steady state, whereas the other one does.
It might be interesting to see whether it can do it, say, with 3 or 4 bacteria.
Okay.
I mean, do they spontaneously specialize? That's the question. Which they should, really.
Yeah, probably.
But it would be nice to see, perhaps.
Yeah. Okay, we will try to implement that. Let me tell you that these simulations, all of these simulations were performed by Saul, who is here, and he optimized the code and parallelized the code and even that, each one of these simulations, the shortest ones take about 1 week. With the 100 bacterial network simulation, it took how? 2 months? 1 month, between 1 and 2 months. So it's not that we can wait forever, okay, because then he has to get a PhD at some point.
True. I'm not saying that.
Max?
Chris. And yeah, just let me remark, because I am also the moderator, that we are a little bit over the time now. People are already getting impatient. People are already getting impatient here. So just a couple of more questions, and then we can ask during the coffee break, okay? And there is this art exhibit outside, please.
Okay, you mentioned that there cannot be complexity or diversity if the mutation rate between host and bacteria populations are the same. So would it be useful or interesting to measure the level of complexity of diversity as a function of this difference between mutation rates?
Well, that's a good suggestion. I haven't thought about it.
Population of bacteria might have different mutation rates, and they might interact in different ways. Can this be done? Is my question.
No, it should. Sure, it can be done, but we haven't done it. But it's an issue to explore. Yeah, of course.
That was my quick question.
Chris?
Yeah. So just, it's a bit of a follow-up to what Francois just said. So, I mean, the thing is that, an arbitrary network can presumably approximate a certain set of functions, but not all functions. So the thing is that what you're emphasizing is the speed of rate of convergence, or of the error. But presumably, what you could do that would be kind of interesting too is to basically say, here's a network, but there's certain things you can't model because it just doesn't have the, it's best like saying, hey, this thing can do cosines, this thing can do sines, but you need both of them together to
nice smooth function or something like that, just to see how selection would act to bring in the other ones, other than just from a rate of convergence point of view. In other words, it's doing a task, well, literally it has something that this thing couldn't get ever, right?
Mm-hmm.
So,
yeah, well, thank you. Well, thank you for coming, for staying. Let's thank the speaker again. Gracias. Señor. Luego te digo. Habla muy mal.
Okay. Let's see. Okay. Yes, yes.
Hello. Okay, let's start the last talk of the afternoon session. Thank you for staying with us until the end of the day. Now, this is Denis Boyer. He's a professor in the Institute of Physics of the university. He has great work. He will tell you about it. So please, Denis.
Thank you, Max, for the nice presentation and for the invitation, obviously. So I'm going to talk about dispersion, meaning the dispersion or diffusion of animals or living organisms, and in ecological context. And this is a work done in collaboration with students, Teodoro Danman and Gabriel Mercado and Octavio Miramontes, who is a researcher that maybe some of you know. And most of these results are presented here in this reference, which is here. And you can even find a link to some kind of vulgarization article about it in Spanish. So that might be of fun to read, although it's not very technical. The field of movement ecology maybe has become important in the last 10 years. So it's a relatively recent area of ecological research, and it is dedicated to the study of movement of animals or living organisms, which can range from bacteria to large animals. And that study movement per se, and can ask very general questions about why organisms need to move, why it is so important for life, how they do that by the locomotion, what kind of energy consumption locomotion how do they manage to move, and where do they move, and when, it's what are the navigation and the pattern of by themselves. Actually, I just mentioned as the journal Movement Ecology, which is an open access journal which has appeared since 2013. And so, for instance, there are a lot of methodology that this topic deal with, because we have to analyze typically movement data. And typically, we can consider this has a lot to do with animal tracking in the wild. And so traditional tracking is just to follow the position, the XY coordinate as a function of time of a tagged animal, for instance, followed by GPS. So I also mentioned in parentheses that that field is also very closely related to human mobility. So because we are not very different from animals in our displacement in many aspects. And also, so we will have a trajectory function of time. There are other things that can be done in more details. For instance, if you put some devices on this animal, like a 3D accelerometer, you could have access to what is it doing actually. Is it staying still, sleeping, or moving, or the head, or jumping, or doing some kind of activity that gives some information on the behavior. Or you can even remote sensing the weather data, what are the climatic condition around that individual at some particular time. And also something which is very difficult to do in ecology, which is to get information about the habitat, the environment through which this animal is going through or living generally. And you can also, for instance,
to understand some interaction with other tagged animals, social interactions, or other kind of ecological interactions. So now the technology starts to exist that would do these things. And from individual trajectories, one can try to understand statistical properties, to analyze them as a physicist would analyze the movement of a Brownian particle, for instance. And to say if there is anything interesting. Of course, we would like to understand what are the underlying ecological processes. I mean, does movement reveal some kind of interaction, of adaptation of the organism with its environment? Is there some particular interaction and cognitive processes that are at play and that can explain what these animal are doing, if it's really completely random or if they use memory or if they are more complex interaction, and what would be the biological function of movement or these kind of patterns of movement that are analyzed. And of course, this is important for applications in biology, for instance, in conservation of ecosystems, and also to understand health issues and disease spread, for instance. And here we are mainly interested in modeling issues, how to model animal movement. The simplest model that exists is, and the most used traditionally, is a diffusion. Like animals are diffusing like a Brownian particle, and which microscopically can be explained by random walk movement. So you have, for instance, a walker on a line that performs some random jumps with a 50% probability to the left or to the right. And as we know, after many steps, so first we can write a master equation for the evolution in time of the probability that the walker is on site n at time t. So that would say, okay, what is the chance that I have to see an animal at that position, given that I knew that it was starting at that position at an earlier time. And we know that this is a well-known Gaussian distribution, which is characterized by a width which grows slowly with time as the square root of time. So the mean displacement here, which would be the average n squared, the deviation around the origin grows as linearly with time. And this is a well-known Einstein-Fick law for diffusion of Brownian particles. So here is an example of a Brownian walk in 2D, so which is a big zoomed here. And it is composed of, I don't remember, but about of 10,000 steps. And we see this kind of movement that actually resemble a lot the movement of real animals when they are looked, they are analyzed at some particular temporal scale. And here we have some other mathematical objects. And in the title, I was talking about anomalous dispersion. And first, it was invented as an abstract object, so which is essentially a random walk like this one, except that the length of the displacement between turns. So here you arrive here and you do a random reorientation. You choose an angle, you choose a length, you perform the length, and so forth. Is distributed according to a scale-free distribution behavior with power law behavior with some exponent beta between 1 and 3. Here, actually, there are also 10,000 steps. Also, they look really much less than the number of steps that are here, because we don't see the many small steps, and the trajectory is completely dominated by very large relocations. And actually, animals sometimes really need to go from one area to another area, using too much time in some part of in some region of space. So these flights are characterized by non-Gaussian distribution and some kind of anomalous transport properties. So in physics, for instance, in turbulent flows or in plasma,
particles tend to diffuse this way rather than this way. That means that the typical displacement square is not linear with time, but is super linear with time. We have an exponent here, alpha larger than 1, and sometimes we call this super diffusion. So it's a fast diffusion. Okay, so as I said, there is many evidence of Levy processes in biology. For instance, in this kind of bugs, seabirds, or in unicellular organisms, in completely different settings, scales, and habitats. So here is the trajectory as a scale. Here is a 10-kilometer scale. Here it's maybe a few microns. Here we see a trajectory obtained by GPS, very precise, of an albatross flying over the sea. Here is a little zoom, a little small part of the trajectory which is zoomed here, and that's revealed a quite rich structure, which in a way resembles the original structure here. So there is a kind of self-similarity which is typical of Levy flight. There is a fractal dimension which is associated to that. And even if we would do another zoom here, probably would see, again, a similar pattern. And this is, so the power law distribution. So something similar happened with these microzooplankton, which are predators. So they look for prey. These prey are other small microorganisms. This guy is looking for fish or krills or these kind of things. And this is kind of qualitatively similar pattern, like a very non-uniform occupation of space. And here, so I talked about these power law distributions for the step lengths, and by plotting in log-log this step length distribution P of L. So more or less, there's some evidence of some power law behavior, which is consistent with a Levy flight rather than with a Brownian movement. So here, here there is an index which is beta equals to 2. So I recall that an interesting property of Levy flights is that the diffusion coefficient is infinite. So this distribution have problems when you deal with second moments or first moments, or the average of L or the average of L squared can be infinite. Well, is infinite in Levy flights, and that gives rise to these interesting mathematical properties. Of course, in the wild, in the real system, it's not really infinite because there is a cutoff, but there are also evidence in many other species, like sharks, tuna, turtles, and small krills, that they are performing in their environment. These are marine predators, or marine prey for the krill, that performing Levy flights. And here is an evidence of this super diffusive behavior on spider monkeys. So this is a Mexican example from the Gabriel Ramos Fernandez, obtained from a study system in Punta Laguna in Yucatan, where we see the super linear growth of the mu square displacement with time. Here, maybe you can see, but there is an exponent 1.7 instead of 1 for Brownian motion. And so there is a handful of examples of similar behavior with exponents that are in general around 2, the beta equals to 2, which is just right in the middle of the Levy regime. And even in a human context, like fishermen, nobody traps, banknotes, and cell phone users were observed to do this kind of anomalous diffusion, rather than random movement with a well-defined diffusion coefficient. So many of these patterns are usually interpreted as search processes. So search process is when something or someone is looking for something or someone. So that happened in many contexts, for instance, in pollination, in foraging, and even in biophysics, where typical, you have an element, like pollen, for instance, that needs to reach other places, other flowers, in order that something happen, like reproduction, or this foraging, that guy is looking for prey, and he doesn't know where food is, and there is a search which can be systematic or can be quite random and irregular. So here, for instance, we have an example of antigens that look to bind by a T cell that would produce, for instance, antibodies. And this takes some time because the entity who is searching needs to get there for the first time. And this time of first arrival is a stochastic element. So this is the most well-known theory that explains why Levy foraging is so ubiquitous in nature. And it is a very simple model, which is a search model, where you have prey randomly distributed in patches, which are these red dots, and you have a single predator that perform randomly oriented steps with this Levy distribution and move at constant speed and capture the prey within a vision radius. So whenever a prey comes within a vision radius like here, it is captured, and one measure, for instance, the number of prey captured per unit of time. And the main finding is that there is a maximum for beta in the Levy regime. Yeah. Yes. Absolutely. Yeah, that's exactly. That's why it is written here that they are distributed in patches, so there is not completely random distribution. Okay, so they are fixed, and as you see, it becomes clearer and clearer that there is a maximum inefficiency here when the density of patches becomes small. So when the system becomes scarce. So it's difficult to find things. And you have to perform kind of many steps before getting to one of these for the first time. And but of course, it is important that there is a non-uniform distribution here. Yeah. The size of patches is not so important. Let's say that the detection radius should be much smaller than the distance between patches. That would be more or less the idea. So there is the Gaussian random walks would be over there. And the ballistic limit is when you go in a systematic way, so there is no randomness in the movement, and you go in straight line. And something in between that seems to be optimal. So Levy patterns are interpreted usually as an optimal random search strategy. So this has excited a lot of people and produced quite a large number of publications. Mainly physicists were very excited, and biologists not so much about the idea, although there are some exceptions. But there are kind of the pro and the against of this theory. So there was many criticism about whether wandering albatross care about math, or how many animals really do the Levy walk, or fish in Levy flight foraging, etc., or which contrasts with anomalous diffusion springs, spread its wings, foraging success recorded in situ, etc. So here the aim here is not to get into this debate and say wrong is right, is to try to understand the problem or the origin of Levy pattern from a very different point of view. And more generally, Levy flights are multiple scale movement because you have a scale-free distribution of length. And what is the relation of this mode of foraging, of movement, with population dynamics? So a very important goal in ecology is to relate processes across scales, in particular, to relate the microscopic scale with a macroscopic environment. So when we look at a single individual, this is like looking at the microscope, very small entities in a whole ecosystems, right? So what is the connection between what is doing and the stability of this world system? So, and all this idea of foraging efficiency has to do with an adaptation of the organism to its environment. So what Williams called in 1966, like organic adaptation. So which is, for instance, typically maximizing fitness or an individual reproductive success or foraging success. But on the other hand, so this is an n equals to 1 problem, where n is the number of entities. And what happens in a large population when you have n much larger than 1? So what might be optimal for n equals to 1 may be very different in the context of n much larger than 1. And this is called sometimes adaptation, where we are focused not on the survival of the individuals, but on the species, on the population abundance. And so we consider many interacting individuals subject to demographic processes, competition, predation, and try to answer the question which role plays individual mobility on the stability of the whole thing. And of course, we want to do that computationally and mathematically. So we need to choose a simple system, a model system. The simplest one is one prey species and one predator species, simple Lotka-Volterra. And we will put special emphasis on systems that are fragile. So that will become clearer maybe later on, but that are close to some extinction transition, an extinction transition, or even several extinction transitions. And so the hypothesis, which may or may not be true always, is that multiple scale movement can have a stabilizing effect on the system which is dangerously threatened by extinction of one of the two species or both. So the setting is that is this word of metapopulation that was introduced by Levins some time ago. So metapopulation is a set of population simply separated in space from each other. So here you have patches of resources. They are separated in patches. Population can be natural because of a natural tendency to aggregate, or because they are produced by human perturbations or human activities that have destroyed this environment. And there is only some small fragment that remain. And this population can be connected if fluxes of individuals exist. And this is very important. That's the main contribution of Levin and of his first model, that occurs through corridors, for instance, greatly improves the overall stability of the system. So these patches need to be connected. Well, if they are connected, it would be much less likely that they disappear because of their own dynamics. For instance, predation, competition, etc. Okay, so we have considered a model which is stochastic Lotka-Volterra model. So where these red dots are the prey, their resource, they do not move, they cannot grow outside their patch, and we have predators that are these small dots that move randomly in the system. And so the predator can die with a rate mu. They can reproduce when they eat a prey with some probability. The prey can also reproduce at some rate. So there are these typical demographic parameter, mu, sigma, lambda, and the diffusion of the predator. So the predator move with some power law step length distribution. So some limits, beta very large is the nearest neighbor. So this is a cellular automata, if you wish, that was first studied by these people. So you move randomly this guy, and if there is a prey, he eats it and he reproduces. He can also die, and etc. So there are very simple rules. And we can see what happens with Brownian predator first. So we have, for instance, evolution time of the total abundance of predator and prey densities. And what you see here is essentially a set of disconnected islands where there is a kind of equilibrium between these prey and predator, which are shown in these number of individual as a function of time curves. And so what happens if we decrease the patch radius or we increase the predator or decrease the predator mortality? So typically an initial condition which would be like this, little by little will lead to state with very few biomass. A lot of patches here have disappeared, and this is an absorbing state. Once the predator patch have disappeared, there is no way by the rule that they can recover. So you have an extinction of many patches, and this guy will also get extinct because they have nothing to eat, right? And maybe a few parts will survive, but maybe not for very long. And so we can see first, so that I think this is exemplified here. So we can see the important an important feature of this fragile system is that the disappearance of patches and also of the predator that need patches to survive. And if, for instance, there is a patch that has been added afterwards, which cannot be colonized again, and it stays there, so the prey are happy, but it's a less diverse system because the predator cannot get there. And we have another kind of extinction that instead that everybody gets extinct, the prey survive very well, and the predator got extinct, and that happens when they are not able to find the patch. So when typically you perform some relocation randomly in the system, so here we choose some systems that are very fragmented. So the density, the area, the volume, the area, the fraction of area covered by the patch is very small. So if you just get by random by random on the screen, it's unlikely that you would be on a patch, and that's basically what they do. They don't, so they die before even the value of the reproductive rate and the value of the mortality rate. There is an imbalance that leads to a predator. And then there is, with exactly the same parameter value, what the Levy predators can achieve. So same patch radius, same mortality reproductive rates, same regeneration rates for the prey, etc. And we have some kind of much more stable steady state, so where nearly all the patch survive, and the abundance of predator is much higher than in the previous case. So this is what we see here is that we have different system where just by varying the movement parameter beta, that goes from highly random movement or ballistic to completely Brownian dynamics, as a maximum. So what is plotted here is a density of predators, the average abundance, gets to a maximum value in from range of parameter beta in the Levy regime, in particular, in this case, around 2. And we see that what is the effect? So this is exactly the same curve for the prey. And what can be seen here is the effect of decreasing, of perturbing the system, decreasing, for instance, the patch radius. So you have a much more fragmented system with fewer resources. And you decrease the predator mortality. So you have some kind of, you facilitate proliferation. So the predator do not die easily. And so once they are on the patch, they will really overexploit it over time and reproduce and be there and reproduce until they completely finish with these resources. Mm-hmm. No, there is no adaptation.
When they found the prey, they can leave.
Yeah, exactly. So there is no memory, and so we'll see what can be the effect of adding this effect. At the end of the talk, I will say a word on this. So we see that decreasing here quite significantly the radius of the patch, as I said, and the predator mortality produce a fairly important decrease in the density and a very small here. And if you change your movement strategy, so if you are more Brownian and not Levy, you will see that perturbation, so this decrease of resources, produce a dramatic decreases of the population of predators that can even go to extinction here. So, so there is a kind of robustness, if you wish, with this Levy strategy. So in order to understand more this problem, it was necessary to make some mathematical mean field theory, which kind of simplifies the model that I just presented, but still captures some important things. So we consider a periodic array. Here you have patches. The predator can jump some patch randomly from one patch to the other with this power law distribution kernel. There is an evolution equation for the population, so reproduction, mortality. This is related to the movement part. So lambda naught is a movement rate. And this is what happened in a prey patch. You have a logistic, you have a carrying capacity for the prey, which cannot exceed the value 1, say, and a reproductive rate. And here is a predation rate. And we can solve this system analytically in the steady state and study the average predator density at very large time in a steady state or equilibrium configuration. And of course, this is an approximation because any noise or stochasticity is neglected in this deterministic approach. All the noise we see in the simulations is not present here, so we assume that in each of these cells there is a large amount of individuals, so that we can neglect all the stochastic stochasticity due to the demographic and movement stochasticity. So, here, we denote as A naught this is a periodic system, so it makes things simpler. And A naught is a density of predator on the prey patch. So what we find is that the average density of predator in the whole system is a logistic function of the density of predator on a prey patch. So there are predators everywhere, but that's the density of predator on the prey patch that has a very simple expression that relates very simply to the total. And we can have, for instance, different scenarios. So these are the expression, and these are this is the part which is due to movement, to random diffusion. And these are all the as a demographic parameter that I mentioned. So, for instance, you can be in this situation where in the absence of movement, you have a certain abundance. And typically what happens, presence of movement will decrease that density. And there are several different scenarios. So if we decrease this density, you will see that the average abundance is a whole system will tend to increase. So there is a beneficial function of movement. There is just an increase of the total abundance of the population. But there are all other situations. So for instance, when you increase the population, but then because of this logistic form, you go through a maximum and then start to decrease again the population. So actually that gives you a definition of what in this context would be a fragile ecosystem. And that explains why the levy would be adequate for this system. Typically here we have two extinction thresholds, zero total abundance. So here, this is an extinction due to end exploitation. So the predator are not able to find the prey. So that's the first, and to eat and to reproduce. And here there are too many. They are au contrary, there is a huge predation pressure on this patch, and the prey go to very low level, and there is a second. This is a different extinction by overexploitation. And here in these scenarios, just by varying one single parameter, a movement parameter, you can bring the system of this point or close to the other point. So, which means that you kind of fine-tune the movement parameter in order to be to some reasonable level of abundances, and there is a maximum. So, which is more or less in agreement with the simulation. And so sometimes the Brownian strategy would be optimum if you are in this case, or the random relocation would be optimum if the parameters are such that they vary and then you have a maximum value. And so in this case, simply the random movement is beneficial because there are too much pressure on the patch, and a little bit of diffusion just improves things. And here there is too much movement, and they go away and they don't find prey. So what we call fragile system is more or less the most fragile system are the first case, and that's why a levy exponent, a levy strategy could be advantageous here. So here, this is some phase diagram showing, depending on low mortality, or whether the mortality of the predator is high or low, or the reproduction is slow or fast, what should be the best strategy to adapt in different cases. So that's the comparison of theory with simulation. So it reproduces more or less the maximum, which is here. So here it becomes very bad because, as I thought, it neglects stochastic fluctuation. And all these curves are completely dominated by noise because the number of individuals becomes very small. And suddenly there is a finite probability that a system got extinct because there is zero copy of predators in the system, which cannot happen in the deterministic. So here is another thing that we can do numerically is to calculate the probability of coexistence between two species in the fragile system. And we see that that probability decay very rapidly, so away from unity, except in a narrow region, which is around here. And at the end, induce some incorporating a little bit more sophisticated movement rule. For instance, site fidelity, because animals have memory and sometimes they like to come back to this place that they know. So we can model this by resetting some at any time with some rate or probability or any particle to inside here. So that avoids the spread by diffusion. And we can see that when plotting the abundance as a function of this resetting or memory parameter, there is a phase transition at a critical value of R, from which a system that would not exist, that would be simply empty of where predator cannot sustain any population, start to rise to a finite numbers. And then when this resetting is too high, start to decrease again because of overexploitation of the patch and the too high predation pressure. So for the conclusion, so I wanted to show by this example that in ecology, mobility plays an essential role on interactions, and in particular, multiple scale motion, like Levy flies, but there are other models, can avoid extinction, maximize abundances, make system resilient in front of external change. So you suddenly change some parameters and see how the system respond to that. In contrast with single scale diffusion or well-mixed system. And in particular, this is critical in fragile systems that are endangered by different, for instance, 2 different extinction points. So in this case, you really need to fine-tune this mobility parameter. And of course, in the future, there are other things in particular that can be done. For instance, consider more realistic systems, not just one predator species and one prey species, but many species related to each other by complex network of trophic interaction. And maybe confirm this more precisely by field study, by laboratory experiments with small systems like microorganisms. And so that's it. Thanks.
Yeah.
Thank you for your talk. Very interesting. You mentioned in future work that you can look at memory effects as well. Mm-hmm. How about effects of aggregation of even the same species? So I know that there are some animals that, like sheep, for example, they go off and they'll graze, but then they'll run back and they'll aggregate. Maybe something scares them or something, and they want to, aggregate for safety. So I think that would probably affect that dynamics of that behavior as well, in addition to memory and these other factors.
Yes, of course. Yeah, actually, that's very relevant question. Actually, we're interested in a system which is very similar to these sheep, analyzing data, not within this framework, but more analyzing single trajectories and see their properties on primate groups, particularly in baboons, that show they are scared of being too far from each other, but they need to be far from each other to forage and to find food for their own. And so there are some kind of dynamics, which is in the data very interesting, and that we try to reproduce by a model. And of course, these are non-social predators, but I mean, we could think about how to implement this to the situation that you mentioned. Yeah.
So I have a question again about adaptation. So I just want to summarize to make sure I understood. So the idea is like when the predator is Levy-flight distributed, there is more hope for the prey because basically they can clear out the prey, the patch, you know. Whereas if it's Brownian, they can they stick around and they eat everything. Exactly. But if you imagine a natural system where you have Levy-flight predators, and they're also adaptive, then they have the best of both worlds. You know, because here the first conclusion was like to say, well, there's a hope. If there's Levy-flight distribution, then they can coexist even if you don't have a lot of prey, for instance, right? But in fact, we know that Levy-flights are better to find patches. So they find them better, and once they find them, if they stick around, then that's the end, right?
Yes.
Yes.
Yeah, actually, we didn't do this model, the simulations with the Levy walk model, but with a Levy flight model, which is kind of slightly different. And here in the Levy flight, you lose completely memory from your previous position, well, except you have a random increment. Whereas in Levy walk, you really go through the path, and you can eat a lot, so everything on the way. So that's something we are trying to implement and to see what are the differences. But there are also other things related with adaptation, but real adaptation, like a change of behavior with time. And actually, if you take these random walks and add some memory and in the proper, with simple rules, you can produce some non-diffusive behavior or localization around resources, which is what many animals really do. But then if you finish the resources, you have to readapt. So on this scale, so the scale of movement is a very short time scale, and the scale of reproduction, predation, much longer. So there's a clear separation of scales in problem. But definitely there are many, there are many interesting issues with adaptive walk because they change their behavior in function of their history and their past experience. And they could do that with very simple rules, which are memory-based, but not very complicated. I mean, so that could be for future to see that here.
Other question? No? Well, I guess we are too tired by now. So thank you, Denny.
Thank you.
And thanks to all for staying with us until the end. Remember that tomorrow we have a special event at the afternoon session. There is the inauguration of an art exhibit, which you can already see around the building. So don't miss it, please.
Your call will be disconnected.
Se desconectará la llamada.